So let's start by talking about the neuroglia. So the neuroglia are the cells that support the central nervous system neurons. So which are the cells that we're going to talk about here? Cells that support and maintain the functionality of the neurons. So they're all surrounding the neurons in the nervous tissue. Astrocytes, microglial cells, ependymal cells, and oligodendrocytes. Okay, so these are all in the central nervous system. So I'm going to show you first the astrocytes. Super cool cells. So the astrocytes, they promote exchange between the capillaries and the neurons. You see how here you have a capillary and here you have a neuron. The yellow is a neuron. Capillaries are around it. So this exchange between the capillaries happens because they allow the astrocytes promote and promote is a good word, capillary permeability. So how permeable these capillaries are going to be, that whatever is in the blood is allowed or not to go in touch with the neurons in the nervous tissue. What is allowed to pass? Everything. Wastes that you're just getting ready to eliminate are going to go to your nervous system and intoxicate your brain? Hopefully not, right? So the astrocytes keep substances that you don't want to go to the brain out of the brain. So what the astrocytes and the capillaries do is what we call blood-brain barrier. This is not a barrier like that you have here or in the spinal cord and it's just a wall and things don't go in. The barrier is everywhere in your central nervous system, promoted by all these astrocytes that are holding the capillaries and saying, you substance you're not going to go through. Oh yes, you are allowed to go through. Alcohol, you can go through. Make that person drunk, right? Yes, because anything that is liposoluble, like alcohol, like some certain types of drugs, can go through the capillaries. They just pass through because this is full of fat, right? So anything that is liposoluble just goes through. And then you get drunk, for example, or you can get high on some specific drug because you went to your brain. Even other drugs that are not necessarily recreational drugs. Any medicine that acts on the nervous system has the ability to transpass this blood-brain barrier promoted by the astrocytes. Okay? Next cell, microglial cells. These cells are, you see, they have a small body and all these projections here. These are similar, they kind of become macrophages and they help to eat debris, wastes that are secreted by neurons and other cells in the nervous system. So they're like a cleaning cells, okay? Ependymal cells. What is the name or how you classify a group of cells that are lined side by side, that have an apical side and a basal side, and are lined in a cavity? This is a cavity. First chance. Alternative A, connective system. Alternative B, epithelial tissue. I said connective tissue or epithelial tissue. Which one do you think it is? Epithelial? Correct. These are epithelial cells with cilia. These cells change in shape between squamous and cuboidal or columnar. I forgot if it was cuboidal or columnar. So they can be like flatter or longer, but they have cilia. What is this cilia doing? We have cavities inside our brain and in the spinal cord. So they line these cavities and make this fluid that we have that is called cerebral spinal fluid flow, and they move. They make that flow as a stream in these cavities. So that's what these cells are there for. So they help with, not allowing substances to go into the brain tissue because, right, the brain tissue is here. So something that is here, the cerebrospinal fluid comes from the blood. It's a filtrate from the blood. So once that fluid is here with substances that you may not want in your brain tissue, they just don't pass because of the cells, the protection that they confer. Other substances like salts and ions that you want them to go through, they're going to be allowed to pass. So it's an epithelium with all the epithelial functions that we know that are possible with a protective layer like this. So these are the ependymal cells. And look at this. I think maybe these are my favorite cells. The oligodendrocytes. They are like those little bears that hug? They're hugging cells. They hug the axon. So let me tell you who is who here. Blue cell, oligodendrocyte. The axon, what is the axon? The axon is the arm of the neuron, right? We're going to look at the neuron in a minute. So the axon here is in yellow. See, there's like a long arm here. And the arm is being hugged by these oligodendrocytes. It's like the cytoplasm with the plasma membrane, right? Like a projection of these oligodendrocytes wrap around the axon. Why? Any idea why? This is fat, right? Plasma membrane, it's fat and water. Fat is going to isolate these axons. Remember that you're going to have action potential, like an electricity current going through the axon. So you have the signal passing from neuron to neuron. So you have the function of the nervous system happening, which is send signals back and forth. So this signal, when it passes and emits this is called myelin sheath. See, myelin sheath. Myelin sheaths are isolating. So when an action potential gets here, it quickly moves here and it has another connection on this dot. And it quickly moves to this dot and it quickly moves to this dot. So any axon that is myelinated is going to have a much quicker propagation of an action potential. Cool? That's what you need to know here. What else? Let's talk about satellite cells. These cells are similar to astrocytes, but they're in the peripheral nervous system. So now we're talking about neuroglia in the peripheral nervous system. Satellite cells and Schwann cells. Schwann cells do the same thing as oligodendrocytes, myelin sheaths. Satellite cells do the same thing as astrocytes, promote that type of barrier to protect the neuron. Okay? Do you have cell bodies in the peripheral nervous system or just axons? Totally. You have the whole neuron out of the central nervous system in the peripheral nervous system. So the whole cell is out of the spinal cord, out of the brain. Okay? And finally, our dear friend, neuron. So the neuron is the guy, right? He is the guy that is doing everything, and all the other cells are just serving it, right? So the neuron has, let's call it 1, 2, 3, 4 important parts that we need to know. So you have dendrites. What are the dendrites? This is the receptive region, okay? It's what's going to receive the signal from another neuron. So just keep that in mind for now. And then the dendrites are connected to the soma or cell body. So you can see this cell body named as soma, S-O-M-A. That's another name for it. This region here, the beginning of the axon, this like triangle region. I hate that my pen is not here, but it's not. I don't know. My computer is kind of crazy these days. I think it's being affected by the pandemic, and I don't know where my pen went. But this is the axon hillock. right? And the axon hillock is the beginning of the axon. And then the axon is all this, all the way here. And this is the axon terminal. Okay, so let's talk about how the impulse is going to go through. Starts in the dendrite, goes to the soma or cell body, starts to get propagated always in this direction. It never goes the other way, okay? See, impulse direction. So then the action potential or the signal goes, and we're going to talk about what this signal is in a minute, okay? Goes through the axon all the way to the axon terminal, and then as an electrical or as a chemical response stimulates the next neuron. Who is the next neuron? It depends on where you are. It depends on if this is really a motor neuron or if you're talking about a sensory neuron. So we're going to talk about these different types of action potential and where they happen soon. Okay? So this is the neuron. What are these little blue balloons? The myelin sheath made by the oligodendrocytes. Okay? So impulse always goes this way. When I say impulse, action potential, signal, synapses. Synapse is what happens here between the axon terminal and the next neuron. But this is all going to promote synapses, right? All right. Now, let's look. This is a neuron. How pretty. Sometimes you can't really tell where is the axon and where are the dendrites unless you know what's the direction of the impulse, okay? It happens that you can't really tell. Okay, so let's see what else I want to tell you from part A. There is a little more detail in the book about how myelination of fibers happen. When I say nerve fiber, I mean the axon, right? And how the Schwann cell in the peripheral nervous system wraps around and make this kind of like roll around the axon. There is an electron micrograph here showing this is myelin sheath. This is like a cross section of your axon. How cool is that? That's an electron micrography, a picture taken by an electron microscope. And so remember, myelin sheath, I'm going to say one more time, is responsible for what we call saltatory. Let me see if I can insert. It's not allowing me to do anything here. So frustrating. No, nothing. Oh damn. Okay, so myelin sheath is going to allow. It's in the book and in the learning objectives. Saltatory conduction, which is this jumpy, leapy conduction. So the action potential is coming this way and then goes boop, 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 all the way to here. And as it jumps, this saltatory means leaping conduction. As it jumps, it makes it quicker. So you have some action potential that happen quicker than graded potential. We're going to learn the difference because the myelinated ones are going to be quicker. You have action potentials that are non-myelinated, and they're still action potentials, but they're going to be slower because they don't have the myelin sheath, okay? Classification of neurons based on their structure. So you have multipolar neurons. Usually neurons that look like this are motor neurons and interneurons. Motor neurons are the ones that are innervating your muscles, right? And interneurons are the ones integrating sensory and motor information in your central nervous system. So these two types of neurons are the ones that we call multipolar. And then we have, they look slightly different even though they're classified both as multipolar. You have bipolar neurons, which are neurons that you find usually in, you see their shape? retina in your eye, and olfactory epithelium. Okay, this is mostly where you find this type of cells. And the unipolar cells that have this head out, the cell body kind of sticks out. So how could you tell here what part is the axon and what part is the dendrite? You couldn't tell unless I tell you that the stimulus starts here and goes to here. So this is the axon terminal, these are the dendrites, okay? You couldn't really tell unless I tell you. But you see peripheral process, so this comes from the dendrites and the signal is going this way to the central nervous system. How do I know? Because it says receptive ending. So receptive has to be from the sensory side. Only the unipolar neurons are sensory neurons, as I said, right? Here's another slide that shows the comparison between them. So again, multipolar, Purkinje cells in the cerebellum, pyramidal cells, which are also in the central nervous system, or motor neurons look like this too, in the peripheral nervous system. Bipolar cells, olfactory or retinal cells look like they're classified by their structure as bipolar cells. And unipolar cells are the ones that you find in the sensory peripheral nervous system, the sensory neurons. This is just showing you how each one is functionally different. So showing you a neuron in the retina, a motor neuron that is a multipolar neuron, sending an impulse to a skeletal muscle or to a visceral muscle. And the sensory information coming, from example, from the skin to the spinal cord. So you can feel something, right? Okay, I think we're done with the first part. Let's see each other again for part 2, where we're going to talk about the resting potential, action potential, greater potential, and so on. I hope this was clear. Bye-bye.